Hello viewers, welcome to this particular lecture on phloem tissue and the food materials conduction through phloem tissue. In this particular lecture, I shall be discussing about the constituents or components of phloem tissue. I shall also be discussing the different means by which food materials are translocated, and here also I shall be mentioning about the different theories that explain this translocation. And of course, I will also be talking about the criticisms or evidences for these theories, and towards the end I shall be stating which particular theory is most acceptable and why. Let us start with the phloem tissue. Actually, in a plant conducting tissue or in a vascular bundle, there are two types of tissue, the xylem tissue and the phloem tissue. The xylem actually helps in the upward conduction of water and dissolved minerals from root to leaves. So there is always upward conduction, and this phenomenon is called ascent of sap. But on the contrary, the phloem tissue is mostly helping in the transport or translocation of sap or food material which is produced in the leaf, and there are different tissue of the plant which gets this food supply from the leaves. So this particular phloem tissue is showing upward, downward or lateral translocation. And when we consider about the phloem tissue, there are different components, but all these tissue are compacted towards the outside of the vascular bundle. It's obvious because the xylem is mostly woody, maximum part of the plant tissue is formed of xylem tissue, but phloem tissue is only very much compressed outside, towards the outside of the particular plant trunk. So when we consider the different components, they are the sieve cell, the sieve tube, the phloem parenchyma, phloem fiber, the companion cell, and of course there are other types of cell which are not really part of phloem tissue but associated with phloem and remaining separated from the phloem and xylem. So they are mixed type of tissue in between phloem and xylem. Now as such, the sieve tube is the most important constituent, and this sieve tube is acting like a continuous channel. The end of this sieve tube is formed of sieve plate, and this sieve plate is actually acting as a sieve which will allow the movement of food or sugar, and if it is big size molecule they will be automatically sieved off. So the sieve plate is very important barricade through which certain toxic materials cannot move in or move out. And they do not have any nucleus. There is a continuous cytoplasmic channel which is actually favoring the movement of food along the phloem tissue. Now this particular sieve tube is living, but at the same time it is non-nucleated. But when we consider about sieve cell, they are living and they are having nucleus, but they are present in lower plant or plants which are not angiospermic. So they are mostly the gymnospermic or the pteridophytes where the sieve cells are present. Along with them, there are two types of cells which are companion cell and albuminous cell, and of course there is the phloem parenchyma. They are basically metabolically active because they are living, having nucleus. They provide the metabolic activity of the phloem tissue, and at the same time they are allowing the transport from the source to sink. There is also another component which is non-living and woody, and they are the phloem fibers. And these phloem fibers basically they bind the outside of the vascular bundle and prevent it from getting totally destroyed, or protection is the most important function. The albuminous cell is somewhat similar to phloem parenchyma, but this albuminous cell are like companion cell accompanying the sieve cell, so they are present in gymnosperms. But phloem parenchyma on the other hand, they are present along that particular companion cell, and they are helping the metabolic activity or movement of sugar or sucrose from sieve tube to some other tissue or components. And when I talk about parenchyma, there will be different types of parenchyma. Phloem parenchyma is a normal parenchyma helping in metabolic activity, but there may be AERENCHYMA where there will be air spaces within, and in addition to normal translocation, they are helping in making the plant lightweight, or they are helping in flotation or buoyancy. At the same time, there will be specific chlorophyllous parenchyma which are present in the mesophyll tissue, which are chlorenchyma, and they have a specialized function of photosynthesis. Now companion cell, as I told you, is associated with the sieve tube controlling their metabolic activity. But this companion cell can be of two types. Just the one which is close to the sieve tube, they are called the ordinary companion cell, helping in translocation. There is the another type which is transfer cell, and the transfer cell are bigger in size. They are in between the xylem and phloem, and they are somewhat contacting or connecting the two types of major tissue in plant system. And as I told you, the phloem fiber is an aggregate of dead tissue, and gradually they are formed from the fusiform initial cell, and gradually they become elongated 5 times, 10 times, 30 times, producing thick fascicle. And these phloem fibers, they protect the vascular bundle, and ideally they are present in the jute fiber or they are present in the jute stem, and they are called commonly the secondary bast fiber, which is having tremendous economic importance in the field of industry or packing industry. Now let us talk about the translocation mechanism. Now the translocation mechanism is actually movement of sugar molecule from source to sink. Now what is the source? The source is the leaf tissue where the glucose is produced, and the glucose is converted to sucrose. It gets into the companion cell and it gradually comes down into the sink, where through companion cell it gets into the lower cell or root. But this continuous movement is helped by water which is moving along the xylem tissue. So directly, though the food is moving along the phloem, the movement of water is basically giving the osmotic pressure through which the translocation of food is possible. So this is basically called the source sink movement mechanism. Now as I told you, the movement of water along the xylem is always in upward direction, because the availability of water is from the soil, and the water moves up into the leaf where photosynthesis occurs, and then the sugar moves down. So definitely through phloem downward translocation is there, but sometimes it so happens there will be bidirectional or multidirectional flow, because what I am trying to say is that from an upper branch there will be movement to the lower branch, or from the outer part to the inner part of the vascular tissue this movement will occur. So that way phloem translocation is dynamic in nature, showing multidirectional movement of sugar. Now let us talk about the active transport of sucrose through phloem tissue. This active transport is helped by proton or H+ ion. Now H+ ion will move out through the membrane, as a result of which this H+ ion will create a gradient outside the membrane. And this particular H+ ion will combine with sucrose, and together there will be co-transport where sugar will move in. So in this way, sucrose and H+ ion together they move in, and once they move in, this is the living membrane of the companion cell, they will be gradually transported into the sieve tube, and in the same way it will be translocated in long distance mechanism through the phloem tissue or through the sieve tube, and will reach the particular sink where it is also received by the companion cell and will be stored in the storage organ. So there is a unique way which is called the symport, where the H+ ion is favoring the movement of sugar molecule along that particular living cell, which is the companion cell in this case of higher plant. But apart from sugar, there is also a unique mechanism by which some other molecules are moving, and thereby there will be a long distance remote control. That is, I will produce a particular messenger RNA, and this messenger RNA will be transported along the phloem tissue, and some other metabolic activity will take place in some other cell. So this is basically, we call it the producer cells in shoot, which is producing the M5C mRNA, and this M5C methylated mRNA will be transported along the phloem tissue, and it will be received in the root, and where this particular mRNA will be functional, producing some specific protein. So it is not that all the time the food is being translocated. We could see here there will be long distance traveling of mRNA, and this is ideal, because whenever there is a grafting, in grafting what happens, the lower part is the stalk, the upper part is the chion. So the chion is producing some particular mRNA which is translocated into the stalk, and there will be some metabolic change, and thereby we see that the stalk is gradually now behaving in the line of the chion. So though there are two different plants, but ultimately it is the chion which will be dominating, and a single plant with single character will be established. Now when I was talking about this mechanism of this mRNA Later on it was found out it is not only the mRNA, the translocation stream is actually favored by different other types of RNA like the miRNA or the siRNA, and this movement also will take place through the companion cell, and this type of mRNA or miRNA or siRNA movement will allow the signal transduction. And this signals transduction will be involved in different types of activity which include infection, which include miRNA transcription, which include transposition. So there are different types of activity which is actually taking place in a distant cell, though the actual miRNA is produced by a different cell. And the transport of this miRNA is facilitated by specific protein like the PSRP1. And this PSRP1 protein will produce a ribonucleoprotein complex which will bind with single-stranded RNA and will allow the long-distance movement of miRNA. And this movement will take place across the living companion cell of the phloem tissue. When we consider the plant, there are actually three different types of movement of RNA that occurs in this particular plant body. And this RNA transport can be normal RNA transport or endogenous RNA transport. It can be the transport of viral RNA. It can also be the transport of the PTGS or post-transcriptional gene silencing. Now let us talk about the first one, that is the long distance transport of endogenous RNA. Here that particular mRNA from companion cell will get into the sieve element like the CM-NACP, and this CM-NACP mRNA will go along the sieve tube. In a distant cell it is expressed and there will be specific protein synthesis. This is occurring in majority of the plants. But when there is systemic transport of the viral RNA, what happens is there is an infection in a particular cell with a particular plant virus, and this particular viral RNA will be transported along the sieve tube and the expression of the disease will occur at some other point, and if there is a resistant gene then the disease cannot be developed. But what I am trying to say is that the virus is invading at one point but the expression of disease takes place at some other point and it is facilitated by the phloem tissue. The third one is the PTGS signaling, where the PTGS is post-transcriptional gene silencing, where the PTGS signal enters the sieve tube and it is translocated along the sieve tube, and finally what happens is this particular expression of the PTGS will allow in the expression of a particular protein and there will be a metabolic change, and this is ideally observed in case of grafting. Of course, there has been some role that along with PTGS there might be some viral RNA moving around, and this viral RNA movement is also, there is some research that heavy metals like cadmium will prevent the expression of this viral RNA or the viral induced disease in plant. Now ideally the function of the sieve tube becomes clear when we consider the component present in the phloem sap, and that include the cyclophilin, the rubisco, the cysteine proteinase, the glycine-rich RNA binding protein, the PRP. So different types of protein components are there which include the photosynthetic machinery also. So what can we say when we do the 2D gel electrophoresis? Different types of protein are distinctly produced inside the phloem tissue, which definitely indicate the dynamic nature of the sieve tube. Based on this, scientists have seen what are the materials that are translocated through phloem, and they include the carbohydrates, which includes the sucrose. So basically, though we produce glucose or the plant produce glucose in photosynthesis, glucose ultimately is translocated in the form of sucrose. But apart from that, there are also other components which are translocated that include amino acid, that include protein, inorganic ions, and of course the plant hormone or phytohormone or growth regulators. And the rate of translocation is pretty high. It has been calculated that 1 meter translocation occurs per hour. There has been specific experiments by which this particular translocation through phloem can be explained. One such experiment is the ringing experiment. In the ringing experiment, what is done, the outer component or the outer phloem is removed. So the xylem being intact, the upward conduction of water and solute molecules will take place and the leaves will be intact. But the food which is produced in the leaf cannot be translocated in the lower part, so the lower part will gradually be withering or getting withered, and thereby there will be some swelling just above the girdling stress. And there has been also another proof by which there is radioactive leveling of water in the leaf. Whenever there is radioactive leveling of water in the leaf being the site of photosynthesis, sugar will be produced in the leaf. And this particular sugar will take that particular radioactive H+. And as a result of which, when we print it, the autoradiographic detection will be there in the phloem tissue, which definitely indicate that phloem is actually the site for sugar translocation. And it is not the xylem, because here the xylem is only allowing the movement or upward movement of water, which is not labeled. Another interesting theory which was denoted by Munch is actually the bulk flow hypothesis. The bulk flow hypothesis ideally explains the movement of water molecule along the xylem and the movement of sugar along the phloem. What is actually done, there is a concentrated solution and a diluted solution in two definite flasks, and they are kept in a particular tumbler and interconnected with side tube. What is happening is, from the concentrated solution gradually there is bulk flow of water and sugar, and thereby they are getting into the dilute solution, and in the same way the water is moving out and they are actually getting inside the first tumbler. So continually this movement of sugar and water takes place between the source and the sink. So this particular theory is definitely showing or explaining the steps of phloem translocation. And what are they? The glucose is produced by photosynthesis in the mesophyll tissue. The glucose is actually utilized in respiration, part of it is converted to non-reducing sugar or sucrose, and normally the sucrose concentration is 10 to 30% in that particular photosynthetic cell, and this sucrose is actively transported along the companion cell into the smallest vein of the leaf. It is diffusing out through the plasmodesmata, reaching the companion cell and finally to the sieve tube. And thereby this particular sucrose movement takes place through the sieve tube, and it is favored by the hydrostatic pressure of the water which is moving along the xylem, and it is interconnected to the root. So thereby we say continuous water flow upward is basically controlling the movement of sugar solution in the leaf. And this particular hydrostatic pressure moves the sucrose and other substrate towards the sink. In the storage sink, the sucrose is removed into the apoplast and it enters into symplast. And the water is moving out of the sieve tube by osmosis, which is gradually reducing the hydrostatic pressure, and the pressure gradient is established. So we thereby see that a particular source is continually producing sugar, which is maintaining the concentration gradient, as a result of which this sugar can be translocated to other different tissue. So ideally Munch's theory is explaining the movement of sugar molecule along with water through phloem tissue. And there are specific evidence of that. One is there will be exudate from the phloem. Whenever there is the outer part removal of the stem, there will be exudate of sugar coming out, which indicate that the sugar is present in the phloem tissue. And at the same time there will be concentration gradient of the organic solute between the root and the leaf. Obviously the leaf will contain more sugar or sucrose. And of course when there is a particular virus moving along the tissue of phloem, that will be definitely from the leaf to the other part of the tissue, if that particular virus has entered the plant through some natural opening. But at the same time there are some specific criticism. And what are these criticism? Mass flow, we could see it is a passive process, and here what we see also, that sieve tube vessels are supported by companion cells, and companion cells are also having the particular ATP. In the same way, amino acids and sugars are also translocated. Along with sugar, amino acids do not have the same rate of translocation. Another interesting criticism is that when we say bulk flow, it is always from the source and sink, so it is unidirectional. But actually phloem may show bidirectional or multidirectional movement of sap. So there are other theories. One such theory is polymer trapping theory. Now what is polymer trapping theory? Here what is said, that in the bundle sheath of the mesophyll, there will be combination of glucose and fructose which will produce sucrose, and this sucrose will enter into intermediary cell, and in that intermediary cell the sucrose will combine with galactose producing raffinose. So raffinose is actually a trisaccharide, and this trisaccharide is moving inside the sieve element, and this trisaccharide or oligosaccharides in this way they are moving along the sieve tube following the concentration gradient. And there are specific mechanism by which phloem loading and unloading takes place, and there are at least 4 different ways by which this phloem movement occurs. One is typically apoplastic loading. Now apoplastic phloem loading mechanism is that the sucrose which is synthesized in mesophyll cells diffuses to vascular parenchyma and then to the apoplast. But in symplastic polymer trapping, the sucrose is mobilized symplastically from mesophyll to the surrounding cells. And in case of symplastic passive loading or diffusion, it is just without any energy, sucrose is translocated. And in revised symplastic passive loading, there will be specific vacuolar transport of sucrose, and it specifically occurs in rice. There is also a specific sucrose gradient that is produced inside the sieve tube, and normally this specific gradient of sucrose from the upper part of the sieve tube will move gradually to the lower side following the concentration gradient, and this does not require any energy. But the most important phenomenon that was discovered was the hair-like phloem protein, which was denoted under electron microscope, and this phloem protein translocation is very interesting because through SDS page at least 2 different types of phloem protein, about 100 kD and about 20 kD molecular weight, they are the PP1 and PP2. And what does these phloem proteins do? The phloem proteins actually help in the translocation of sugar. And as I said, the PP1 is the primary structural protein which is present inside the phloem, and PP2 is acetyl glucosamine binding lectin, which is covalently linked with other protein by disulfide linkage. And basically the PP1 and PP2, they are showing contractile movement, and this contractile movement helps in gradual translocation of sugar. And in addition, PP2 is more dynamic because PP2 has the capacity to interact with the mesophyll plasmodesmata, which will increase the size exclusion from cell to cell. And at the same time, PP2 also can bind with microRNA or messenger RNA, which definitely indicates that the PP2 or the phloem protein is mediating the transport of the RNA, which is helping in protein synthesis in a distant cell. So thereby we can conclude that the translocation of food takes place through phloem, and this particular mechanism is explained through different techniques. There are different theories which explain, like the bulk flow hypothesis, the contractile protein hypothesis, and thereby we say that the phloem protein is the most important constituent inside the phloem cytoplasm, which is allowing the movement of sucrose or other sugar inside the sieve tube. Thank you.